Hello, my name is Kathleen Antonakakis, and I'm going to be presenting my project entitled Comparison of Bacterial Growth Absorbed Through an Artificial Epidermis in Organic versus Non-Organic Cosmetics. You may be wondering what the exact difference is between organic cosmetics and non-organic cosmetics. Organic cosmetics generally is associated with protection of nature despite the creation of new products. There are no genetically modified organisms, herbicides, synthetic fertilizers, or artificially manufactured chemicals because these are considered harmful to nature. On the contrary, non-organic cosmetics has the potential of containing harmful ingredients, including artificially manufactured chemicals. One commonly used chemical is preservatives, intended to prevent microbial decomposition or any undesirable chemical changes. Parabens are a type of preservative which are found in everyday toiletries like cosmetics. In organic chemistry, there are many types of parabens. However, after doing research, I found that the displayed parabens are very common in cosmetics and were especially present for my experiment. It included methylparaben, ethylparaben, propylparaben, isobutylparaben, and butylparaben. The problem is that parabens have been connected to hazardous side effects, including disruption of hormone development, breast cancer, reproductive problems, and allergic reactions. These are all due to parabens' tendency to be absorbed through the skin very quickly. The specific quality of parabens makes me question whether they are effective in preventing bacterial growth, especially after being absorbed through an artificial epidermis. After other research and study findings, such as those located in the middle, I found that the demand for organic products has been on the rise for young millennials and Generation Z. Regardless, companies' obsession over marketing has caused confusion to common consumers. Companies may claim that an item is organic simply to increase their gross domestic product, a tactic in order to gain favor in the competitive cosmetic industry. Constraints have additionally been developed to prevent false advertising, such as the USDA seal and the NSF seal located on the left. However, it is not always easy to read the fine print seen right and determine the potential harm in each ingredient. It is hard not to question chemicals' presence in cosmetics in the first place, especially if they operate ineffectively. Human skin is generally divided into 3 layers. The hypodermis on the very bottom serves as fat storage. The middle layer is called the dermis, containing nerve endings, sweat glands, oil glands, hair follicles, and blood vessels. Finally, the epidermis is the outermost layer consisting of the acid mantle, stratum corneum, and various pores. Both the acid mantle and stratum corneum together are expected to operate as a defense against microbes, although many people are unaware of the fact that poor maintenance of the skin can alter these mechanisms. Constant application of products, especially containing chemicals, can lead to weaker barriers and easier absorption through the epidermis. This results at times in bacteria slipping between the lipids and corneocytes and settling into pores. When initially researching for this experiment, it was difficult trying to find an artificial skin that embodied at least some of the characteristics of human skin. Soon enough, I found Jenna Wong and Clarissa Young on a Hong Kong TED Talk displaying their wishes to market their own artificial skin called Happia, the letters each referring to a characteristic of real human skin that the artificial skin is able to embody. The product as a whole was intended to help victims with skin loss, as Wong and Young had described a powder explosion that had occurred in Taiwan. It included a hydrophobic water barrier, hydrogel, and calcium chloride layer combined together to stimulate regeneration after skin loss. They discussed how human skin has the ability to prevent bacterial growth, and they incorporated their waterproof layer and licorice root extract to ensure that Happia did the same exact thing. For this particular experiment, I was only observing bacterial growth in the case that a product would slip past the waterproof layer. As a result, only the hydrogel portion was recreated in order to replicate the epidermis. Wong and Young chose specific ingredients to ensure that the homogeneous hydrogel could absorb materials. This included the use of sodium alginate, xanthan gum, and liquid calcium chloride seen here. From this point on in experimenting, I assumed the artificial epidermis would absorb as Wong and Young had discussed. The bacterial growth would be measurable after sampling, no contamination would occur with the use of isopropyl alcohol, and because the products chosen were used before, this would be easy for testing the effectiveness of parabens stopping bacterial growth. The alternative hypothesis predicted that bacterial growth from non-organic products would be the same or greater than that of organic products due to the parabens' potential to absorb through the skin quicker. The null hypothesis simply states that growth from non-organic products would be less, which is expected from companies. This experimental design contains the basic variables, constants, and trials. The independent variable, dependent variable, and constants are straightforward. For clarity with the trials, there were 23 samples of the artificial epidermis in order to correspond to the 22 products chosen plus 1 control. Next, there were 2 trials overall. The first trial contained 23 agar plates for sampling of the 22 products from pre-absorption, plus 1 control. And the second trial contained another 23 agar plates for sampling of the 23 epidermal samples, including the control. For step 1, the products were sampled. A cotton swab was used to take the sample directly from the product and transfer it to an agar plate. All the agar plates were then kept in the incubator at approximately 40 degrees Celsius for 4 days, a time determined to allow for full growth of bacterial colonies, yet no drying out of the agar. After this period of time, the number of colonies on each plate was calculated. For step 2, several prototypes were made, following Wang and Young's video demonstration. The final formula included 5 grams of sodium alginate, 15 grams of xanthan gum, and 160 milliliters of distilled water. Step 3 was to assemble the artificial epidermal samples. This is where the calcium chloride was utilized to prevent sticking of samples. I made this flowchart and repeated it for each sample made. For step 4, the products were actually applied to the epidermal samples just made. This process was also repetitive in applying 0.1 grams of each of the 22 products to the epidermal samples, leaving 1 as a control. They were carefully applied and placed into the incubator at 37 degrees Celsius, which is body temperature, for 4 days. Finally, for step 5, the epidermal samples were taken post-absorption for sampling. 1 epidermal sample was taken at a time and cut utilizing scissors. Then cotton swabs were used to obtain a sample from the inner center part of the artificial epidermis, because the experiment calls for testing absorption through the skin. For this reason, taking a swab only from the top of the sample would not be sufficient. All of the agar plates were then kept in the incubator at approximately 40 degrees Celsius for 4 days. After this period of time, the number of colonies on each plate was calculated. Directly after choosing the cosmetic products that were going to be utilized, this frequency chart was generated to display the parabens in the 11 varying textured products and how frequent they would appear. It could be seen that propylparaben has the highest frequency with 90% and methylparaben was the second highest with 73%. After assembling the artificial epidermis, a caliper was used to determine the thickness of each sample. I recorded this simply because I had a feeling that these numbers would vary. With the thicknesses determined, the standard deviation was found to be greater than 0. For future improvement of this project, I would use a custom 3D printed stencil instead of a simple plastic stencil, allowing for a more consistent thickness among the epidermal samples. Located here are pictures of the artificial epidermis samples, before absorption on the left, after application in the middle, and after absorption on the right. These pictures show the agar plates pre-absorption and post-absorption. As seen in the photos, bacterial colonies produced before absorption were present, yet they were fewer in comparison to those of after absorption. Those produced from after absorption were small yet more numerous throughout the entire agar plate. From those agar plates seen before, the number of bacterial colonies was counted and simply placed into this chart. This scatterplot helped in comparing the number of bacterial colonies before and after absorption. The circles represent products containing parabens, whereas the squares represent those that are paraben-free. It simply helps in seeing the growth from before absorption in the product and after absorption through the artificial epidermis. Finally, I calculated the percent increases from before and after absorption. In 7 out of the 11 cases, the percent increases of the paraben-free products were greater than those by parabens present. On the other hand, the remaining 4 cases demonstrated a larger percent increase from the products that did contain parabens, the textures being liquid pore filling, stick medium coverage, liquid full coverage, and pressed glitter. All these textures were of some form of a liquid substance. To complete this study, a t-test was formulated comparing the percent increases. In the first t-test, this included the percent increases of all 11 textures in 22 products. The p-value derived was 0.265485. It was greater than the significant value 0.05, being one of many indications showing that the null hypothesis could not be rejected. A second t-test was then formulated, only consisting of the 4 textures that had higher percent increases from the products containing parabens. The p-value of this second t-test was 0.013399, which was lower than 0.05, indicating a rejection of the null hypothesis. To conclude, when found in a liquefied texture, parabens do not work efficiently in preventing microbial growth. As a result, bacterial growth occurs in the product, and this bacteria is ultimately absorbed into the epidermis. This provides an explanation to one of the paraben's known side effects, allergic reactions. Bacteria burrowing into the epidermal pores has great potential for skin irritation. Paraben's inefficiency in a liquid texture can be explained through simple principles of skincare, in which specific nutrients and ingredients are formulated in a specific texture for maximum or minimum efficiency. In liquid textures, parabens simply do not operate as they should. Therefore, use of parabens in any cosmetic product should be considered for termination in future cosmetic items, simply because we should remember that they are chemicals and there are still many unknowns that remain. Finally, for future studies, I wish to identify the bacteria obtained either with the gram stain test, bacterial transformation with P-glow, or differential media. Additionally, I would work more with observing expiration dates, and lastly, I would attempt to expand the collection of products to testers found in various beauty shops. Thank you for listening in, and I hope you enjoyed.